Hello everybody, I am Nikita Kohli. I am a master's student in the Master of Data Science program at Thomson Rivers University. Today I am going to talk about a new workflow Pentapen which combines penalized models for the SNP identification on whole genome Arabidopsis thaliana data. Genome-wide association studies, abbreviated as GWAS, aim to identify the relationship between genetic variations and physical traits. Single nucleotide polymorphisms or SNPs are the most abundant form of common simple DNA variants. Since the whole genome SNP data are typically high-dimensional, detection of significant SNPs is challenging. Feature selection algorithms based on statistical and machine learning methods are often used to tackle the high dimensionality problem. This research aims to tackle this challenge by proposing a new workflow, Pentapen, which combines 5 penalized models: Ridge, Lasso, ElasticNet, Group Lasso, and Sparse Group Lasso. It enhances the chance of identifying potentially significant SNPs by leveraging the properties of feature selection from the 5 penalized models. In this study, the data from a model plant, Arabidopsis thaliana, is used to test the performance of the proposed workflow Pentapen. This genome data has 1307 samples with 214051 SNPs or features in terms of statistical parlance. 3 phenotypes from the phenotype data were selected, including binary, continuous, and categorical phenotypes. Anthocyanin is the binary phenotype which maps the presence or absence of anthocyanin. Width is the continuous phenotype which indicates the plant diameter, and germination days is the categorical phenotype which gives the number of days to germinate. The study evaluates binary phenotype by using classification performance metrics including precision, recall, F1 score, and AUC. We evaluate regression models using R-squared and root mean squared error, in short RMSE, and multi-class classification models using accuracy for continuous and categorical phenotypes respectively. As the genome data may have varied format, it is important to process the raw data to make it compatible with the penalized models used. The nucleotides in the genotype file are recoded to numeric values to form a binary marker data. A design matrix of dimensions p times n is created to make it easier for performing mathematical operations and transformations in high-dimensional marker data. This is followed by forming a transposed matrix of dimensions n times p. The null values are removed from the input phenotype data and are matched with marker or genotype data. The resulting data after the pre-processing are then split into training and testing sets. 80% and 20% of data is used for training and testing respectively. Five-fold cross-validation was used to train the models. In Pentapen, first all the SNPs of whole genome SNP data are utilized to train Ridge, Lasso, and ElasticNet. The union of the output SNPs from these three models is selected, and we call this step as SNP pooling. Secondly, the selected SNPs are sent to Group Lasso and SGL. The union of the output SNPs from the two is the final output of the Pentapen. Finally, an aggregated model is developed by combining the predictions of all 5 penalized models. This model accounts for the strengths of each individual model while reducing the impact of their weaknesses. This aggregated model was used to calculate the performance matrix of Pentapen. The final output of SNPs from Pentapen are used for SNP validation. The SNPs are validated using the Arabidopsis Information Resource website to find genes and genes functions corresponding to the SNPs. The final SNPs are also compared with the SNPs output from a GWAS software GLBIO. Pentapen is implemented across 5 folds parallelly, so there are 2 outputs obtained from each penalized models while training the workflow. First, important SNPs, and second, the prediction for the phenotype. These results are interpreted to confirm the superiority of Pentapen by comparing the performance of the workflow against all 5 penalized models using all SNPs, the computational time with every single model, and the number of SNPs selected by each penalized model. Therefore, from figure 2 on the poster, it is noted that for binary phenotype, Pentapen outperforms group lasso and SGL but performs similarly to ridge, lasso, and elastic net. For continuous and categorical phenotypes whose results are not displayed here, it was evident that Pentapen outperforms ridge, group lasso, and SGL while still performing similarly to lasso and elastic net. Further, compared to the individual penalized models, Pentapen requires a longer execution time. The performance matrix show similar values between Pentapen and single penalized model, and the computation time is a bit longer for Pentapen but still within the acceptable range. The advance of Pentapen is that it selects reduced number of significant SNPs which are easier to assess further for biological interpretations. SNP validation indicated that some genes found based on these SNPs from Pentapen showed functions contributing to the phenotypes. A shared gene AT3G08970 found between GLBIO and Pentapen for the binary phenotype increases the confidence in the gene's association with the phenotype. This gene is found to compensate for growth defect in the Arabidopsis species. We conclude that Pentapen can be used as a complement to GWAS as they use different models, for example, generalized or mixed linear models in GLBIO versus penalized models in Pentapen. Finally, there are 2 potential future works that researchers may focus on. First, we suggest combining Pentapen and GWAS software, which may help to identify more potential genes associated with the phenotype. Second, we may study how the model differences between GWAS and penalized models influence the varied SNPs identified from them. Thank you for listening to my presentation.